Hello everyone. Welcome to this seminar on organoid technology and organoid chip technology. This is the first seminar given by Hub and MIMETAS together. We've worked together for a number of years already to develop our combined technologies. And now we think it's a good time to share with you what we've been doing over these years. Basically, the idea is that we tell you on the one hand about the Hub organoid technology. I will do that. Robert Vries, by the way, my name is CEO of Hub, followed by a presentation of MIMETAS on their organoid chip model systems, and then basically to tell you how we can combine these two technologies in order to improve the models, of course, but also to allow us to make more complex model systems that we cannot do when we take organoids in a dish in itself, or when we, and that is a very important point of this as well, when we use specific cell lines as opposed to patient-derived model systems. So the two presentations will be following each other. If you have any questions during the presentation, please type them into the chat box, and we will answer them in the Q&A session directly after this presentation. Also, if you're watching this online later on demand, or if you just have questions later, please email them to us and we will get back to you offline. So as said, I will start with this with the presentation, a presentation on the Hub organoids. What we would like to get into, but as in specific for this seminar, is that when we are developing model systems, these newer model systems, the model systems, the next generation model systems, as we like to call them, what we need to consider, what you need to consider in the lab is, on the one hand, always the cells that are used, the biology. That can be an animal model, that can be a HeLa cell, if we take two ends of the spectrum. Organoids, our organoid models, for instance, adult stem cell derived, patient derived organoids, iPS technology. Those are all the cell lines, the cells and the biology that you have to choose from before picking a model that you think can answer your questions. On the other side of that, we have the physical environment in which we ask these questions, and that's also just as important in getting the right answers. And that is, am I using a Petri dish, 384 well plates, ultra low attachment plates, or nowadays the more technologically advanced organ-on-a-chip models, in which we can put our cells in a much different conformation so that they mimic a normal organ or organ physiology or even disease physiology in a much better way. So I think that is the message from this seminar, and what we are trying to convey to you is that nowadays, when we have the much more advanced, I think, model systems than we had maybe 15, 20 years ago, we can really try to combine the best of these two technologies together in order to get a good answer to the questions that we have in the lab. Organoid technology, as you can see on this slide, it is basically a continuation of why also we make organoids with organ-on-a-chip. But model systems, we're working on them now in this way for a very specific reason, actually. And that is that over the last maybe 30, 40 years, drug development has taken an enormous leap. If you take cancer, for example, we started out with simple chemotherapies, and before that, obviously, only surgical answers to that disease, and have developed that into targeted therapy, small molecules, antibodies, and now immune therapies and even cell therapies that can be used to treat it. That has generated, of course, a lot of new curative therapies for cancer and also other diseases. Nevertheless, we see that still a majority, over 90% of the drugs that we develop in the lab fail in the clinic, and that for diseases like cancer, unfortunately, more than half of these patients still have a prognosis, a very poor prognosis, or a prognosis that it cannot be cured. Part of that is that although we have these very advanced drugs, the models that we've used to develop them on are an animal, a mouse, a traditional cell line, which basically is completely different from the patient that we are trying to treat in the clinic. Because of this, the answers that we get in this development phase are not the answers that we're going to get in the clinic, and the result is that we can cure cancer in mice very efficiently. Mice do not have to suffer from cancer anymore, but humans are still suffering from it and dying from these type of diseases. So what we think is, if we develop a model system that's much more closer to people, then we can get answers there. We can develop drugs that are geared towards people, and we can have a much better result in the clinic, and we understand more of these systems than we do with these traditional models. Hub organoids that were developed in the group of Hans Klevers in Utrecht are a part of that answer, we think, and that is actually the development started from a fundamental study of stem cells. In the group of Hans Klevers, we studied stem cells for a long time, and the idea and what they found was that in the intestine, and then later also in other organs, a very specific cell type in the epithelium, that is the stem cell, the LGR5 positive cell, we call it. That's the stem cell of that epithelium, and that is true for the intestine, that is true for the colon, but it's true for basically any epithelial system that we have in the body. And that was a key finding, because for the first time now we had an organ from which we understood the physiology to such an extent we could point to a stem cell and see how that stem cell developed in the organ, how it took on a normal function in these organs, but also how it developed into diseases like cancer. So when Nick Barker in Hans' group had identified that stem cell, what then Toshiro Sato in his group did, is, okay, now we have this stem cell, can we take it out, can we put it in this, and can we expand it to study it? And then our key drive was, can we take this stem cell, can we grow it in a dish, but not like a HeLa cell that changes to the environment of the lab, but can we maintain its normal physiology, can we maintain its genetics as it had in a patient? And that worked, of course. That's why we have organoid technology now. So what Toshiro Sato did is that he could, with the method that he developed, he could take out single stem cells, or nowadays, just for ease of use, a tissue fragment. We take these fragments or cells, we put them in a dish, and when we expand that in this particular medium, according to these methods, we can indeed expand these stem cells. And then we get a genetically stable culture, and that's the key point. So we see that the genetics of the cell don't change; they are stable in culture over a very long time. In fact, when we talk about the intestine, it's indefinite, and then allowing us to basically study these stem cells in the lab. What was another key feature, a little bit accidental, but was that when we expanded these stem cells, we found that we did not just get a dish full of stem cells, but with this right method, it actually formed an epithelial structure, and that's what you can see also in this slide here. So you see from the left top corner the single stem cell, but when it expands, it forms an epithelial structure where the luminal side of the intestine, in this case, is on the inside, and the basal membrane is on the outside. It's a polarized membrane. It has a normal barrier functions closed, and it has the stem cells, but also the differentiated cells of that system. So what we get, and that's why we call it an organoid, it looks a lot like an organ, is a model system that is genetically stable and also functionally stable, and basically mimics what an organ does, and we can expand it indefinitely in the lab. Another important feature is that, so it started in the colon and the intestine, and that, as said, the stem cell characteristics were similar in other organs, and so was the organoid culture. So although every organ has its specific features in generating the organoids from it, the life expectancy of the cultures is slightly different. Nevertheless, the basics of the systems are all the same, and therefore we could generate these organ models from all these different organs. And last very important feature, it doesn't work just accidentally 1 in 1,000 patients, but it works for the majority of samples that we take from the hospital. So we can take samples from, especially when the starting organ, the intestine, for any patient that comes into the clinic, we can generate these systems and generate organoids from it. So with that, this is actually what was the start of the foundation of Hub, the company starting on this organoid technology. What we realized is that with this system, we all of a sudden had an enormous variety of possibilities to use it. And one of the things that we do, we have listed here the sort of the general directions of how organoid technology is used. But the key aspect is that that we can generate these in vitro patient populations, biobanks, from the different healthy or diseased samples, and that creates a collection that we can use as a virtual patient population in the lab to do our clinical studies. Hub does that. We also actually in an important collaboration with the called HCMI, Human Cancer Models Initiative, where the more cancer models are generated and distributed through the ADCC. It's very important we realize that through technologies as with MIMETAS, but also through collaborations as with the ADCC, we need to make sure that we have these characterized models available to researchers. And so with these biobanks, we basically can create individual patient models with a specific genetic or clinical background that we want to study, or use it for patient stratification to look at a larger group of samples, a larger group of patients to study diseases that we're interested in. That leads to one of the major points, of course, for Hub is the drug development and drug discovery efforts that we can use. It is a platform that can be used as we do with the cell line platform, as for drug screening, etc. And another important point, I will put that in this presentation just to highlight it. But the key aspect of why we wanted to do this, why we wanted to develop a new model system, was that we wanted a model that was more like a patient. So an important point for us always to demonstrate is that is indeed that model then much more like a patient. So we do this by comparing the organoid responses in the lab to the clinical responses of the patient in the clinic of the matching samples. And indeed, what we see is that these are correlating very strongly, and therefore we are confident that what we are looking for in the lab, in fact, is what we can expect to happen in a patient as well. So what in this said, it's a short presentation. What I wanted to show you is a new set of data that was actually generated in the Clevers group. I think very interesting on the combination of organoids with bacterial systems, and then and some data that we generated in the hub on assays that we can make with the organoid models. But first, the one point because that is still the key point of an organoid model, or I think any next generation model. Is the model a patient, yes or no? And what we do is, it is depicted here in this slide, for example, in breast cancer. Here we have taken, this is a paper from Sachs in some years ago already, taking breast cancer samples of a metastasis, we grow it into an organoid in the lab, and this organoid we then characterize. What we then subsequently do, left bottom corner, is we test the drugs that are used in the clinic. And we find in green cells organoids that are responding well to those drugs, and we find patient samples, of course, organoids that do not. And then when we compare that to the data of the clinic, to the actual patient records of the patient that we made the organoids from, we indeed see that the patient in green was responsive to the treatment, whereas the patient in red was not responsive to the treatment. And that gives us the confidence that we are indeed using a model system that tells us much more of what is eventually going to happen with our data and what is the real physiology of patients in the clinic. And that is summarized here. So traditionally we have molecular genetic data the last 30, 40 years, I guess, that has told us a lot more of the patient on top of the clinical data that we've used for since the Greeks. That combination is what we've been using so far to generate our model system, or to generate our understanding behind the biology of diseases. And what we can do now is we add an organoid model to this, which is basically a functional system that can test whatever we want to know on these models. And that combined gives us the understanding of patients, but also allows us to proactively maybe diagnose patients on the benefit of specific treatments. So, as said, two experimental approaches that I want to show you, and before we go to the presentation of MIMETAS on the organoplates. First is this video, which shows, I think, a very interesting study done in the Clevers group. As said, we have the intestinal organoid model that we can use to study different diseases. We had used it initially as a purely epithelial system, but of course bacteria are a very important aspect of intestinal physiology as well. And what the Clevers group did is that they took a specific E. coli, one that expresses colibactin, a common molecule that is produced by bacteria in the intestine, and one they created the coli without it. So this Colibactin expressing population is a population that exists in a minority of patients, but a significant population. And so what the Clevers group did is that they expanded these two E. coli populations and injected them into the organoids, and see that, and to basically have the different molecules produced by the bacteria have its effect on the organoids. They used antibiotics to remove the E. coli, and they repeated this in a couple of cycles lasting up to a couple of months. So basically what we have is an expanding organoid culture that maintains this physiology that we know from a normal intestinal epithelium, and constantly expose it to these different populations with and without Colibactin over a long period of time. And then what they did is we can grow organoids, of course, from single cells. So basically we can take, after months, these organoids that we have collected, that we have generated, grow them out from single cells and make clonal organoid lines and sequence it. And what they then found is very interesting. So they had these two populations exposed to the specific E. coli with the Colibactin and without, and they found that the population that was under the influence of the Colibactin producing E. coli had a very different mutational signature, or had a very strong mutational signature for a specific mutational signature. And that is basically meaning that that basically showed that we have a bacteria that can affect the intestinal epithelium in a very specific mutation, has a very specific genetic toxicity on the epithelium. And in fact, when they looked in more detail at this mutation they found that it creates oncogenic mutations in this. And APC, of course, a major cause of colon cancer, can be caused by the introduction of a stop codon caused by this Colibactin. So I think this is the first time that we have actually shown a physiological, a mechanistic explanation for the role of bacteria in intestinal cancer, which we have thought of for a very long time. But indeed, now mechanistically we can show that indeed, when we take this bacteria with these particular molecules, we can create these mutations in the genome of the epithelium. What is, and going to already maybe to the MIMETAS presentation, what you see is we had to inject the E. coli into this system, and then we got this very nice answer given by the organoids. But what we want to do, of course, is create a system that makes that much more easier and much more easy to handle, to add bacteria and other species to its immune system later on. And this is what organoplates can do. We can there create an epithelial barrier, as I wanted to show you in the last couple of slides, that actually can be used to study this, as opposed to trying to inject every individual organoid over a period of months. So this is the last slides, the last 2-3 slides, which is where indeed, so we can make all the different cells, you see that here, of the intestine. And what we can do with this, we can create in the transwell system, for instance, a barrier, an epithelial barrier, and not in the cystic organoid shape, but in a 2D shape that has the same physiology as a normal intestine. And this is something that with the organoplates, of course, can be done much more specific and much more technologically advanced. And what we can use these type of systems for is that, I'll give you one example here, is that we see mimicking, for instance, inflammatory diseases, that when we, in the left graph, when we take these barriers, when we take these transwell systems, we can expose them to cytokines, as happens in an inflammatory, like inflammatory bowel disease. And what we see is that we lose the barrier, as happens in an inflamed intestine. Importantly, when we use molecules like tofacitinib, a JAK inhibitor, we see that this cytokine-induced barrier function loss is alleviated. So indeed, we see that the barrier function is restored with this drug, as this drug is obviously intended to do that in inflammatory bowel disease. So what we see is that we can use organoids indeed to study this physiology, these disease models, and use it for studying more complex diseases. So the last slide here, indeed we have an organoid, the point of an organoid being that we have a patient-specific model system that can be used preclinically. We can use it in very early phase characterization of diseases, we can use it for genetic characterization, but then in subsequent preclinical development, we can use it for understanding mechanism, for drug screening, and candidate selection. Because we have a whole population of it, a biobank, we can actually use it for patient stratification and biomarker identification as well. And what I think is particularly interesting is that because it is patient-derived and you can create it from basically any patient, it actually supports both the clinical trials and understanding what patients do and do not respond there, or actually mimicking part of these clinical trials preclinically in a stratification experiment. As well as that it shows that patients can or cannot benefit from specific treatments by taking patient-derived organoids and testing preclinically what drug might or might not be helping these particular patient groups. So that is the part from organoids that I want to tell you, and then here we continue with the presentation from MIMETAS, who will explain how we can actually use these different model systems in a more advanced plate technology and to study the physiology of human diseases. So then to answer some of your questions, you see one of the questions that we got was a relatively general question is why should we be working with organoids? What does organoid bring that other model systems don't? I think that one of the comparison that's often made is 2 and 3D. I think that is not necessarily the most important point. The key point of an organoid is that it is genetically stable and patient-like. So we have always used HeLa cells. Myself, when I was in the lab before, we used cell lines. That was the only tool that we had to study human physiology, human diseases. Knowing that the cells that we were testing are obviously not the cells of the patient that we are interested in and have actually changed to the environment of the lab. And that is the one thing that organoids can do. Organoids are as the patient. They are not a human as a whole, but they are the epithelium in itself of that particular patient without changes. So it has all the genetics, and we know it can build part of the physiology. And this is also where, that is the key point. An organoid is, and we can show this much more representing the clinical physiology of a patient than these traditional cell lines do. Why I made that initial point on 2 and 3D is also, and why do we try to make organoplates, for instance? The fact that an organoid is 3D is just as a matter of fact, that happened when we were growing them. And that is very useful for studying many aspects of it, because when we test drugs, we don't need to have a very complex system. We can just grow them like this, expose them to the drugs, and we get, when we look at the clinical comparison, physiologically relevant data. That being said, for instance, the example of the bacteria that I showed earlier on, there are many disease models or many different studies we would like to do where we want different shapes, different mechanistic answers to the model systems that we have, and in which case we want to grow them we want to grow them together with an immune system, etc. So this is where the physics of the system become important. So what from our perspective is the important point on organoids, and so the biology is that you need a genetically stable patient-like system. And then the next question you ask is, can I just grow that as a 3D spheric system? Do I need an organoplate, or do I need other systems in order to ask my specific biological questions? So, and then going to the next question that came in is the challenges that we see in organoid research, challenges, opportunities, often always very close, of course. What we've seen so far, maybe that's the best way of describing the challenge, is that the organoids initially are a relatively complex system. What we have is a model that is so close to patients and that is a good thing and a bad thing, obviously combining the opportunity and challenge. We have a system that is like a patient, but if I do something wrong with it, use the wrong reagents, leave it in the incubator on Friday and come back on Monday, and simple things like that, it changes. It is no longer the patient that I had. It is actually now turned into this a model system that has adapted to whatever I did with my reagents in the lab. So what we need to do, and this is also, for instance, why we have this collaboration with ATCC, why we have the collaboration with MIMETAS and other academic groups as well, is we need highly characterized models. That is the challenge what we need to do. We can now grow with organoids, with IPS, with our modern technology. We can grow all these different patient models, but they are each of them are individual patients, and we need to make sure that when we compare the results of lab A and lab B, that we are indeed realizing that we are either working on the same organoid under the same conditions, or we're working on two patients that clinically might also be very different. So we need to make a conceptual change, realizing that in the lab we need to be constantly vigilant into maintaining our system as it should be maintained, so that we keep the characteristics as they were. And two, that we have, when we compare results between labs, that we compare how did we grow them, are we comparing the same patient, etc. Understanding that we are now really looking at a patient group and not at a HeLa cell that is clonal over the, well, actually it was not very clonal, but has been considered to be relatively the same all over the world. Okay, then indeed going on to other questions, I see that an important point indeed is the recommendations is a question. What are our recommendations on how to use the model system in the lab? There, I think that it is a bit in line with the previous question, that the key point of using organoids, it is considered to be a bit more expensive, which is true, but I think especially if we compare the readouts and the relevance of the readout, I'm not so sure if it is in the end more expensive. It's definitely not more expensive than an animal model. It's much cheaper than an animal model. And the answers are therefore are much more representative than a cell line. Therefore, I think the cost consideration nowadays, and maybe not 10 years ago, but nowadays, I think shouldn't be there anymore. But what is an important point is the reagents. So we need, with these type of systems, make sure that you have the right reagents, that we keep a stable model in the lab, that you freeze it and thaw it, and it comes back as you froze it, that when you grow it for a couple of months, that it's still the same as what you started with. And so this vigilance in maintaining a system, that is the key point. That is also the one thing as a researcher, when you're starting it, you should be careful with. So get the right protocols. We can provide them. They are online on many different sites available. Get the reagents that are in our publications and in other people's publications. And don't, because we get most of the questions we get are when things go wrong, we can take back to actually the use of reagents or use of protocols that didn't allow the expansion of organoids as we think they should when we want to expand them stably.
Welcome everyone. My name is Kinga Kosim, and I'd like to talk about the Organ-on-a-Chip platform developed by MIMETAS and how we can use it in combination with hub organoid technology. To briefly introduce myself, I have master's degree in biotechnology, and during my studies I had the opportunity to work at the universities in Poland, Denmark and Italy. Before starting my PhD training, I was part of EMBL. There I worked on the establishment of genome editing technology to study hematopoiesis process in the stem cell model. My journey with organ-on-a-chip technology started 4 years ago after I joined IT and Mimic Network as the PhD student. During my project, I worked on establishment and validation of an in vitro model for Crohn's disease. And currently I'm working as a scientist at MIMETAS, and I'm focusing on gut modeling in vitro, specifically on the development of intestinal organoid model. In this webinar, I'll discuss the limitations of the currently available models, introduce the organoplate platform, and show two models we developed: tubular intestinal organoids and vascularized tissue on a chip. Modeling human biology in a physiologically relevant manner is very often challenging, and currently available models pose certain limitations. Standard tissue culture methods provide static environment. However, in vivo, many cell types are surrounded by moving fluids. Application of media flow introduces shear stress, which in turn influences the cell morphology and behavior. Most of the microfluidic platforms allow for modeling this dynamic environment. Limitation in most of these more physiologically relevant models is the presence of an artificial membrane. Having a physical obstacle between the chambers of microfluidic device reduces the permeability and consequently decreases sensitivity of the assays. And depending on the material used, it might cause sticking of the compounds to its surface. Besides, the membrane might obstruct the imaging. Establishing a physiologically relevant model is challenging on its own, and doing it in a cost-efficient manner makes it even more difficult. The throughput of the system is crucial in screenings. High volumes of used compounds elevate the costs of experiments. Compatibility with standard lab equipment, the ease of use and possibility of automation are important factors when choosing the system. Speaking of 3D models specifically, in the static conditions, the diffusion through those biological structures is impaired. There is also a limited possibility of establishment of the models that fully reflect the tissue complexity. Lastly, vascularization of said models is very difficult in vitro settings. To overcome mentioned limitations, we developed the organoplate platform. It's a microfluidic device that allows for 3D cell culture with continuous perfusion. Each chip contains a couple of channels, which makes it ideal for co-culture experiments. The platform is membrane-free and allows for good quality imaging thanks to the microscopy-grade glass bottom. The design of the plate is based on the standard microtiter plate format, and that makes it compatible with regular lab equipment, including the possibility to use it in automated processes. It doesn't require pumping systems as media perfusion is gravity-driven. We offer a few types of organoplates that differ in the architecture of the chips. Here you can see a 2-lane and a 3-lane device, as well as an organoplate graft. And depending on the design, each plate contains 40 to 96 individual chips. Our platform allows for development of a variety of models representing unique biology. Barrier tissues can be grown in tubular structures. Introduction of diverse cell types in a single chip is possible thanks to extracellular matrix patterning. Spheroids, organoids, and tissue explants can be vascularized in the organoplate graft. Various immunocompetent models have been developed where immune cells can freely move across ECM and microfluidic channels. As an example of how we combined the hub organoids technology with our microfluidic device, I'd like to show you the primary intestinal organoids grown as tubular structures. This model has been developed in a 3-lane organoplate. The organoids are seated in the top channel, where they form a barrier supported by the ECM in the middle channel. The media is added to both top and bottom channel, and the plate is kept on a rocking platform, which facilitates polarization of the cells. Here we show the expression of brush border markers, ezrin and vilin. Cells form tight junctions, as represented by Zoan staining. A huge advantage of using primary material to model gut biology is presence of various cell types. And here you can appreciate the staining for mucin, which is the marker for goblet cells, and lysozyme expressed in Paneth cells. In standard culture conditions, organoids are grown as spheric structures with impaired access to the apical side. As shown in this 3D reconstruction of confocal images, the organoids cultured in the organoplate form fully developed tubule, allowing for an easy access to both apical and basolateral side of the structure. Having the cells grown in such tubular structure against an ECM allows for assessment of the barrier function, which is a key aspect in toxicity and efficacy studies. It can be used as a quality control measure for the development of the cultures. It can be done easily with a fluorescence-based barrier integrity assay. Fluorescently labeled dye is added into the top channel of the organoplate that contains the tubule. Time-lapse imaging is performed immediately after that, and the signal ratio between the top and the middle channel can be calculated. When no cells are in the chip, the dye immediately flows through the gel and reaches the bottom channel. If the cells formed liquid tubule, no dye passes through and the fluorescence signal is detected in the top channel only. Another way of evaluating barrier integrity of the tubules is to look at the transepithelial electrical resistance. We've developed a device compatible with the organoplate, the organotear. The electrode board is placed on top of the organoplate such that the electrodes fit into channels of each chip. The measurement of an entire plate takes about 1 minute. And apart from permeability studies, molecular transport across the cell membrane can be measured in our system, as the expression of various drug transporters have been confirmed. Tubular intestinal organoids model finds its application in ADME field. We've shown that the tubular organoids express functional CYP3A4. This enzyme is essential for the metabolism of many drugs, influencing their bioavailability. Here we compared the functionality of this cytochrome in Caco2 and intestinal organoids grown in a 3-lane organoplate. We were able to measure the activity of native enzyme and efficiently inhibit it with ritonavir in both models. We observed a dose-dependent, efficient induction of CYP3A4 activity with vitamin D only in the organoids model. And this clearly shows one of the advantages of the primary material-derived model. Another platform that I'd like to present today is the organoplate graft. It's the first cell culture platform that allows for vascularization of the tissues like spheroids, organoids, and tumors. In each chip, there are 2 endothelial vessels that are formed against an extracellular matrix gel. These vessels develop a vascular network under stimulation of a gradient of angiogenic factors. Tissue placed onto this bed is connected to the system of blood vessels, achieving in vitro vascularization. This uniquely facilitates perfusion of the tissue and allows for drug administration through the vasculature. In this time-lapse video, you can observe the formation of vascular bed around a liver spheroid. Organoplate graft is a very flexible device. Various tissues can be placed in a graft chamber and co-culture with many other cell types can be established. In addition to already shown liver, spheroid, and organoids, a range of tumor were successfully cultured in this setup. Our platform, in combination with hub organoids technology, opens new doors for complex tissue and disease modeling and has a great potential for early drug discovery and development. Features like possibility of vascularization of 3D tissues, as well as access to both apical and basal side of tubuloids, allow for generation of more physiologically relevant models. Compatibility of this technology with standard equipment makes it easy to adapt in the lab, and hopefully the applications shown today will inspire your research. Thank you very much for listening. And let me take a look at some questions. Is it possible to co-culture organoid tubules with other cells in the gut or immune cells? Yes, definitely. Since there are 3 channels available, other cell types can be cultured either as embedded in the gel in the middle channel, or in the bottom channel in the form of tubule, like blood vessel, or in suspension like immune cells. You could also think of adding fibroblasts embedded in the gel. What depth into the spheroid can you image to? Are there any size, shape restrictions on the tissue that can be added? Our spheroids are approximately 300 micrometers in depth, and with proper clearing it is possible to see through the whole structure. The chamber itself is 1 millimeter in diameter, so this size is a limiting factor. Are tissue-specific endothelial cells usable for vascularization in the organoplate graft? I'd say yes, it should be possible. However, maybe not all will be able to form a vascular bed, and the optimization for each cell type would be required. What is the long-term viability of the models shown here? For the gut organoids, we kept them in culture for a month in a 3-lane organoplate, and the liver organoids up to 21 days in the graft with stable function. But probably it's possible to grow them longer. I'm afraid we ran out of time, so we will answer all the remaining questions via email. Thank you.